Alternate splicing is arguably one of the most complex processes inside the cells. There are so many different factors which can influence this outcome. So DNA molecule is basically a blueprint for the cell to create a variety of protein products that it needs to do its day-to-day function. This blueprint produces something called RNA molecules, which ultimately give rise to protein products. So what splicing allows the cells to do is to remove the pieces of information from the RNA molecule which is not required to create proteins, and then join together the pieces which are actually required. Alternative splicing allows the cells to cut and join these pieces of RNA molecule in a variety of places. So what this allows the cell to do is to create different protein products from the same blueprint. And this is a very important thing to study, because if this process goes wrong, it can lead to a variety of different diseases. The most important thing is to first understand how this process works. There are a lot of different kind of protein molecules which are known to be involved in alternative splicing, and one such class of proteins are known as the hnRNPs. But it is not exactly clear that how do they actually perform their function. The approach that we decided to take was to try and purify these proteins from human cells and see what other proteins come down with that. What we interestingly found that this protein interacts with another protein called SETD2, which is involved in the process of transcription. And what was striking was that some of the amino acids in the SETD2 protein which binds these hnRNPs are mutated in a variety of cancers. And we believe that more such protein interactions like the one we are finding between SETD2 and hnRNPs exist to make sure that this coupling happens and happens very effectively. So our findings provide very important information on how alternate splicing occurs inside the cells, which is very important to generate the diversity in proteins that the cell needs for the complexity of an organism like human beings.